I'm here on behalf of the Standards Working Group. A number of the other folks who've been involved contributing to this are in the audience as well. We've been assessing the question of where does it make sense to worry about standards in this project, and what are the priorities of addressing some of these possible different places. So the way that we've organized this is, first of all, we are looking at standards from a technical point of view, like data formats and representations and things like that, not from an organizational point of view. So I'm not saying like standards and practices. We also are approaching this from a perspective of, to the greatest degree possible, can we leverage prior work? Never reinvent the wheel if we can. And at the same time, we also think it's very important to avoid premature commitment, either by attempting to standardize something that we don't understand our needs well yet, or premature commitment in terms of not choosing a standard and ending up wrapped around some sort of poor solution just because that got baked in the pilot projects and now it's too expensive to try to change it and we're going to pay for it for the rest of the project. In approaching this, we made a few basic assumptions about the nature of the project. We're assuming that there's going to be a lot of different groups with a lot of different types of roles that need to exchange information, often across radical differences of expertise and goals. We also are assuming, even though we've got the one first project identified, that over its course, this consortium is likely going to have people pursuing multiple simultaneous goals that we want to support off of the same technological base. And that over the course of at least 10 years, we're going to see some major technology changes, including, we're predicting that we're going to shift from a world where right now things are being dominated by synthesis cost towards a world where things are being dominated by yield problems in making and putting together large things, into a world where we are being dominated by challenges of design. In approaching this assessment, we looked at what were all the other working groups talking about and doing, looked at all of the different stakeholder organizations and what are some of the sorts of roles and expertises and goals that they have enunciated with regards to this, and looked at key prior projects like Recoli, SC 2.0, and what's going on with a lot of the big public repositories out there like AdGene, iGEM, NCBI, et cetera. What are the good and bads there, and talk to some of the people involved with some of those. From all this essentially comes our master diagram of our current best understanding of what workflows in this project are likely to look like at an abstract level, and where are the interface points which are likely to be the points of friction where standards are going to be needed. So the interior track here is essentially where we are now in terms of genome writing projects. The exterior track is as we get towards more genome design projects at a higher level, sort of model-driven, parts-driven design. You're starting with a quadrant of you go from a dream into a gene sequence of some sort. Down in the build space, you somehow need to go from a sequence in your computer into actual big modified chunks of strains, up to some scale. At that point, you need to verify that the thing you've built is what you intended it to be, both genotypically and phenotypically, to whatever level of depth people need to go to. And then from what you've learned there, you've got a verification process or a modeling process, or what is the thing you do when you digest the data that comes out of your tests. And we expect that this is going to be a many layers iterative project, just like it has been for other things like Recoli and SC 2.0. You're not going to have one turn around this wheel in order to get your entire new genome, but you're going to go around it many different times, building and integrating. Every one of these these white or black bubbles here is a point where we see an interface where something may cross between an organizational boundary or a tooling boundary. The white ones are digital objects like designs or data. The black ones are physical objects like nucleic acid strands, large constructs, or strains. And in the middle, we also see there are a whole bunch of different databases that are going to be needed, from sort of the simple starting off points where sequences, particular nucleic acid samples or strain samples, up to the data that comes out of running things against these. Then further on, models, and of course, we want to be able to publish these things, share them in journals, and maybe even recover supplementary data and be able to pull it into things that we want to do again. So looking at all of these different artifacts and interfaces, we have come up with a detailed list of recommendations for how to approach each of these interfaces. And we essentially categorize things into 4 groups. Adopt is there is something out there already that we believe solves this problem, and we should use it rather than reinventing the wheel. Extend is there's something out there that almost solves this problem, and with some relatively minor modifications, we think that it could do what this project needs. Create is this is a problem that we are going to have near term that needs to be addressed, and we are not aware of a good solution that can be adopted for it. And monitor essentially means that we think this is going to be a problem in the future, but not now. And so, for near-term, project proposals and such, probably don't need to focus on that. So I'm not going to go through these in detail, except to say that at a high level, we essentially see that in the world of representation of the data objects, from design, inventory, experimental data, there are things that are pretty close to what we need, and we just need to figure out how to adopt them and do the minor extensions that are needed to get them from subgenome scale to full genome scale. In the world of sort of modeling and such, that's pushed off for later. A place where there is a much bigger challenge is thinking about the physical repositories, and the really key challenge there actually comes to essentially linking with what needs to go on with the LC and IP groups. So those are problems that we are not thinking that we should solve, but we want to make sure that the solutions that are getting put together for talking about IP, for talking about privacy, et cetera, are things that have a machine-readable and machine-composable approach to understanding them, so that as this project is operating at scale, you don't have to have a lawyer involved in enabling transactions, and so that you can automatically reason about, I took a piece from this group and I took a piece from that group and I put them together, and what are the IP rights and the legal restrictions on the composite. These sorts of things have been worked out fairly well for digital objects in the past, and so we think that this sort of thing ought to be able to be doable for this project as well. Where we're going from here is, with this sort of core set of recommendations, we want to expand and refine that, hoping for a public release of that sometime in a June-ish time frame. And we think it's time to start forming some partnerships and seeing if we can get initial pilot work in this actually underway in partnership with some of the specific labs. And I'll just close by saying standards are most valuable when they are open and involve as many stakeholders as possible for everyone, so everybody is invited to participate and contribute in these processes. With that, I'd like to invite our panel members up. So I'm going to have Bryan Bartley from BBN, Jonathan Karr, and Leslie Mitchell. I'm just going to leave this up on the screen as a potential talking point. And I actually wanted to start off our discussion on the panel by tossing the ball to Leslie. Leslie was not involved in the things that we put up here, so any mistakes are not her fault, but ours. But we thought for this discussion on the panel, we really wanted to have somebody who has actually been building things at genome scale to start reflecting on, what looks sane and what doesn't, and what we've put forward.
Well, great. Thanks so much, Jake, for the invitation to be here. So I think you guys got a lot right, and dare I say, almost everything right. I think your outline here really captures the workflow. So I think that moving from the SC2 world into mammalian scale genome writing, from the standards perspective is really going to come down to flexibility on the standards imposed. So for SC2, as you all have heard a little bit about at this point, the design was centralized, and I think that generally the project has been incredibly successful, to Jeff Schloss's point, that we have a really good understanding of the primary sequence of the yeast genome. It's complete, and we understand the annotation, so it becomes fairly easy to make edits with predictable outcomes. So to that point, I think having a complete sequence is pretty valuable. Of course, the assembly was outsourced to groups around the world, and the measurement of the resulting products and cells was also carried out by those groups. That information all tracked back and was, coordinated within the network. And so I think all of that was simple, relatively speaking, from the perspective of yeast being manageable. So now tracking up to the human genome in terms of flexibility of the standards we impose, I think as an example, we just need to be able to account for what we don't know now, which is to say knowledge about the primary sequence and the evolving technology development as we go. So one very specific example is the idea of when we design to be able to decouple that design, which could be carried out now, from assembly. And assembly is going to track back to what we know with respect to technology and how we plan to put DNA together and deliver it to cell lines. I think another point would be to ultimately accommodate many different designs. While there's now one primary goal of the project, of course, I think there'll be a lot of other people wanting to do new and different things on smaller and larger scales immediately, so going beyond the ultra-safe cell line. And finally, versioning control on not only the designs, but also the living products that come out of them. So those are some thoughts on what you guys have put together.
Awesome. Thank you so much. Sure. I'd like to give a chance for the other panelists to comment as well. Jonathan, you want to say thoughts?
Sure. So I don't have direct experience in engineering large genomes. My group works on modeling genomes. And so I was excited to participate in the working group because we'd love to see our work in modeling genomes be used to help design genomes in the future.
So one of the questions that I wanted to bring up in that space is, so in our initial assessment, we basically sort of said models are scary and sort of pushed them towards the future. I suspect you might have a different opinion there.
Well, I would use a different word than scary, but they're certainly in the future, I think much further than the timeline of this project. So I think it's important to think about how models could be used for design, but not realistic that in the next 5 to 10 years that will really be possible at a deep level.
Where do you think, so given some of the things that were said today about the initial goals, let's say, project number one, the recoding project, do you see a role for genomic models in that in the near term, even if it's not at a super deep level?
I think there are some areas where it's possible to build genome scale models, particularly metabolism. With that exception, it's quite hard to build genome scale models today. There are, just like GP Wright, there are a huge number of challenges, all of which are unaddressed and for which there's largely no community addressing them. So for that reason, I think it's just not realistic today that genome design can really be motivated by physical mechanistic models in the same way that planes or cars or bridges or electronic circuits are designed today. But we want to make that future possible. I think just like GP Wright is 30 years behind GP Read, maybe GP Model will be at least 10, 20 years behind that. But we want to work to that future.
Cool. Bryan, did you have comments you wanted to add on your perspective on all this?
So I'm also very glad to be here. My name's Bryan. As part of my graduate work, I helped develop the synthetic biology open language, which is a standard for designs in synthetic biology. And one of the most interesting things about working on that project was not just the synthetic biology, but at a lower level, at sort of the bioinformatics level and the building computational tools, it's how you develop a flexible standard or an extensible standard. So, scientists are nonconformists, we're innovators, and you don't want to constrain a community and lock them into, you must conform to this standard. So one of the, nice advancements in computational standards is the ability to have extensible ontologies and data models, which allows you to communicate broadly across the consortium as well as customize it to your particular needs. So if you have some, in-house workflow for DNA assembly and you want to be able to track that, all the steps that you did, and be able to reproduce that workflow, you can encode that information, and you can also share that information with others. And you may not want to reveal all the details of, all your steps that are involved, but you do want to be able to exchange the information that, this was done in so-and-so's lab. At a high level, they did DNA assembly, and, this was the verified sequence that they got. So one of the things that we can help with as the Standards Working Group is helping stakeholders develop and customize these existing standards to meet your specific needs.
I think that the Joint Genome Institute has actually been doing something very much like that internally with their Boost tool, where they've essentially been using SBAL and extending in order to do exactly this type of decoupling you were talking about, Leslie, between design of the primary sequence versus the assembly technologies that they're bringing to bear and the particular plans for how to put it together. Now, they're working on a much smaller scale. They're not doing whole chromosomes, but that may be an example that we can look to. Let's turn it up to the rest of the audience for questions as well. Thank you, folks.
I come from a journal, and so I like the emphasis on shareability, but I was just wondering how you're envisaging, you have a federated approach to this sharing. So does that take account of sustained availability? Because at Science we would normally require deposition in a public repository. But that's very challenging, of course, because the repositories are not set up to deal with data they've never seen before. So I'm just interested in how you're approaching that.
So I think this question of the tension between federation versus sustainment is a really important one, and that's why some things have ended up in very centralized databases. An important thing about how we're trying to approach this in the set of recommendations we're making is to make those recommendations architecture independent from an organizational perspective. Let me take a step into an area that may seem unrelated for a moment. In the computer science world, software development world, there's been this massive revolution over the past several years as people have moved from centralized repository models like Subversion into decentralized repository models like Git. Now, everybody uses Git in a centralized way. They always pick a server, and everybody kind of, organizationally. But you're no longer technologically tied to it, and you can migrate that or, shift. I think that's the approach that we'd want to think about as a community towards this question of federation and organization, which is that we want to make sure that we pick technologies that allow us to migrate data and responsibilities organizationally, even while they're not compelling us to do that prematurely. Because the place where you really get in trouble is if you say, our technology means we have to be centralized, and now somebody ends up essentially, capturing that as an organization. Now, an example of where this has been used is, one of the repository systems that has gotten developed in the SBAL community. It's two different ones: the ICE repository that the Joint Bioenergy Institute has developed, and the SynBioHub repository that Newcastle and University of Utah have developed. Both of those have actually a journal publication model where these repositories federate, and you can have a data collection which is being held privately, put it into the review process of a journal. ACS Synthetic Biology supports this, for example. And then the journal, with its repository, can essentially import it into its archival management and make it public as the article is published. So these things can be very readily supported. That's one of the reasons we put journals on our map as a place for archival management of data over the long run. Did anybody else want to come? I've talked too long. More questions from the audience?
Hi. I think one of the things that I've been sort of kicking around in my head while I've been listening to you guys talk about increasing stakeholder connection and sharing, and the idea of, what is the sort of thing that might happen when you get, two different things from two different places and the IP involved. I know that the BioBricks Foundation started an initiative for an open MTA. Is that something that you think could apply to some of this?
I would be very interested in the lawyers on the IP group analyzing this. We also had a conversation, both the standards group and the IP group had a conversation with John Wilbanks from Science Commons, which had also worked on something very similar. The Science Commons was a project of the Creative Commons Foundation to essentially try to extend that copyright licensing world into a patent world. And it didn't get a huge amount of uptake for essentially a bunch of sort of quagmire reasons, and people not being enough of the right stakeholders motivated enough to make it work. I think a lot of these things really will only work when we start trying to actually use them and finding out what are the realities of where the friction points are, which are a little bit different from the theory. Another of the questions that came up in MTA, so another person that we involved in this and provided some wonderful information was Joanne Kamens from AdGene, and she talked about how since they ship all over the world, they essentially have to have a special MTA regime for dealing with every single different legal jurisdiction that they ship to. And that was actually a massive amount of not just investment on their part, but ongoing maintenance cost. Because, of course, the regulatory regimes and rules are shifting, too. If we can try to build on some of their experience rather than reinventing it, that will be very valuable. Another of the things that AdGene's experience I think we may want to pay attention to, one of the things they pointed us at was the challenge of simply validating that a vector is what you think it is once you're maintaining it in a repository above a certain size. They apparently are very, very picky about dealing with large constructs because they find it much harder to get them a monoclonal sample to maintain. And I don't understand the technicalities of that, but I think, again, we are not the first people to deal with some of these things, and some of the lessons that repositories have for us are likely to be very valuable.
Sort of following up on that, I kind of wanted to pose a question to members of the audience as well. At what scale does it become really important and necessary to be able to go back and access your DNA assembly fragments? So I kind of assume that oligos are, they're cheap, and so once you synthesize your gene, you don't really need them anymore. But I imagine that at a certain scale, you're going to want to hold on to those chunks and mega chunks in case you might need to go back and correct errors, or perhaps it's going to be useful later on down the road in SC 3.0. So I'm kind of curious, at what scale is it really important to be able to logistically maintain those collections of large DNA samples?
Thoughts, Leslie?
I have a few thoughts. So you did reference some of the ontology of SC2. We build starting with oligos, going to building blocks, which are about 750 base pairs, mini chunks, which are 3 to 5 kb, chunks, which are 10 kb, and mega chunks, which are 30 to 60 kb. So up to the chunk stage, the 10 kb stage, that's all maintained in E. coli in the lab. And I would say for SC2, that would be the point where we store DNA. That said, some teams chose to build just mini chunks and then go straight to yeast, so in their case, they would just have a bit more. That said, if I was thinking about GP Write, I would probably have my construct assembly organism as yeast. And maybe then I'd be storing megabase length DNA, and it would be obviously more efficient to store larger pieces in one strain. In that case, if you did want to make changes, you could easily come and edit in yeast with CRISPR, such that it's going to depend on length scales of what you're building. In this case, it's going to be big, so I would say in the 100 kb to megabase range is valuable.
So you'd feel comfortable to throw away everything smaller than that and just rebuild it if you need it again?
Living in New York City, absolutely. I just think it's unmanageable to keep all of that material. I also think building 100 kb should be routine if we're really going to get into this project.
Jeff.
I'll also say that despite the fact that we not only saved all of those things and moved them from Baltimore to New York and then moved them 4 times, internally, the number of times that we've gone back to the small DNAs could probably count on one hand. So we've basically been generating an enormous amount of CO2 from fossil fuel to run our -80s for all of these years. And I totally agree that that black cylinder on the left there could be almost entirely eliminated by the strategy that Leslie outlined.
That would be lovely. One thing I will note that might want to be considered there, and I don't know, I'd be interested to hear how you dealt with this in SC 2.0. One of the things that Joanne from AdGene brought up was that for them, there's a huge difference between thinking about a strain versus thinking about just a piece of DNA from a shipping and regulatory perspective. And I'm wondering if you found that that boundary significant in SC 2.0 as well.
Guess not. So even with respect to SC2, we technically don't really need to preserve the chunks at this point because we have the completed chromosomes powering growth of yeast cells. So in principle, we could be down to 16 freezer tubes at the end of this project. So that'll probably happen. No, I'm kidding. Jeff's a pack rat.
One other question, I think we're running out of time here, is what have we missed? What are friction points in this sort of project that aren't on our map up here?
I think that one immense friction project, friction point, which is only going to arise most likely once additional GP Wright type mammalian projects get underway in some great initiative, is the measurement. So I think much like SC2, I think the ultra-safe cell line is really about making a bunch of silent changes and having something that sort of looks and acts the same but has these new features that make it genetically more flexible or better to work with. And so from that perspective, the measurement, I think, can be pretty well standardized. It will require some sort of new technology and software to support it, as you pointed out. But I think it's doable. It's probably going to be some RNA-seq. It's going to be some sequencing, like genomic sequencing. And those probably are going to be the two measurements, unless there's any other gross morphology defects of the cells. But once you get into, as an example, transplanting human DNA into mouse cells, you've got different measurements to make, and you need to go through development. And so I think that is something to project out in the long term that's not specific to the ultra-safe cell line.
And I'll note, even for the right now, that one of the things on our map is, so if you're validating that you haven't changed the phenotype, we need some way of specifying how much counts as not changing, because there's no such thing as getting precisely the same numbers out of two assays. So that's one of the things that's on our list as a create right now.
Actually, I guess the software developer in me is concerned about only testing a couple of phenotypes. So like typically we find if we only run a couple of unit tests, there are many other errors that we just don't know about by only testing a couple of features of our models. How much are you concerned about not identifying errors early by only assaying gross morphology, growth rate, and RNA expression?
I totally see that point. I think on some level you may have to pick your battles in this project and spend your money wisely. And maybe the money is better spent on doing those same tests in two separate labs by two separate groups and keeping it to a small number of tests. Again, though, this would be some of the high-level stuff could be applied across projects, but specific to the ultra-safe cell line, I do see it possible to institute standardized tests that anyone could carry out in like very SOP-style format.
This also sounds like a potential point where contact with the models might be very valuable because, I'm thinking that if, even if we're just starting with a metabolic model, if you could say essentially, what fraction of the metabolic pathway is being exercised by a particular assay, and then have, a few standard ones that you could consider that can at least give you some information on much of the metabolism.
Also, to just follow up, I think that SC2 had frequent and simple phenotypic testing and less frequent but much more rigorous phenotypic testing. And so to impose sort of stepwise or like size length phenotypic testing protocols makes a lot of sense. Like when you finish a megabase, you would do a metabolomics profile. But if you've only got 100 kb in there, you're probably just going to do an RNA-seq or what have you.
Yeah, that's very similar to how we think about testing large models. It's not practical to test detailed phenotypes at the level of the entire model, but you can certainly do that with pieces, and then you build up to integration tests.
And of course, as we learn more about how much these changes really affect the fitness, we'll refine everything I've just said. Asterisk.
So I think we've got a lot of good directions. I want to thank all of the panelists and move on to the next group. Thank you all.